Hello, botanists. How are we doing? Yeah, can we hear it for Serpentine? Yeah, that's right. So, really happy to begin our session. This is a session that Marina and I organize. We have several exciting talks. Marina, do you want to say hi to everyone? That's Marina, great serpentinologist. So today we will take you on an epic journey from the humblest microbes growing in the cracks of the serpentine rocks to the vast, expansive serpentine landscapes and the wild plants and lichens that grow there. Did I miss anyone's topic? I think that's it. I think that covered it. Oh, and fire. Classic ecological process. Yeah, let's hear it for fire. Was that a boo? We have some hecklers. That's okay. We came prepared for this kind of thing. We know how it goes. So I'm going to kick things off with the first talk. I'm Jesse Miller, and I'm going to talk about the ancient question of how many serpentine endemic plants does California really have? This question has vexed scientists for untold millennia, going back to the era of Galileo and Darwin. But finally, we've reached an answer in the year 2022, and I'm happy to tell you what that answer is. But first, a little background, of course. So we may have some uninitiated serpentinites here in the room today. For anyone who's not familiar, serpentine, it's actually California's state mineral. I see some excitement about that out there. Glad to see it. It's a very special kind of rock that's toxic to most plants. It's low in macronutrients that plants need. It's high in heavy metals, and has a high magnesium to calcium ratio, which also makes life difficult for plants. So because of this, a lot of plants don't do well on serpentine, and a specialized flora has evolved on serpentine in California and other parts of the world, which is one of the most intriguing parts of the California flora, in my professional opinion. And California is distributed pretty wide. Serpentine is distributed pretty widely in California. You can see there's some really large patches in the north, but there are smaller patches all over the state. Some of these patches are in places that are pretty hard to get to. We used to say you have to drive to the end of the longest gravel road on whatever forest you're on to get to some of these patches, which may be one reason why we're continuing to see new discoveries of new serpentine plants, even in the year 2022. And so even though serpentine only covers about 2% of California by area, it's a disproportionate part of our flora relative to that area. And so the specialized serpentine flora that's evolved over the years is a substantial part of California's endemic flora, which is, of course, one thing that makes California really special. Hugh Safford and colleagues did an analysis of the serpentine endemic flora in California back in 2005, and they determined that serpentine makes up 12.5% of California's endemic plant flora. So our goal was to update this previous database that Safford and colleagues had constructed. And so to do this, we updated the taxonomy from the original database based on the big changes that happened when the new Jepson manual came out. As any of the old-timers here know, some of the big and beloved families were split, and other taxonomic changes happened. The youngins may not recall the breadth that Liliaceae used to have in times gone by, but it was expansive. And then we also tried to incorporate, some rustling in the crowd there. We also tried to incorporate new discoveries that had happened since 2005, which there were quite a few of, it turned out, and other taxonomic updates. So we first just updated the nomenclature from the original Safford et al. Paper, but then we consulted with a whole bunch of botanists from all across California to ask them to review the original list and suggest improvements or changes based on their own field experience. And several people who contributed to this process are here in the room today, so thanks to all of you for your help. This really was a community effort. California is such a big and diverse state. You really need a lot of botanists to figure out the story of all these diverse serpentine plants. But in addition to getting input from many botanists, we looked at herbarium records, and we also looked at the primary literature, especially for newly described plants, to try to classify plants based on their affinity for serpentine soils. So as in the original study, we ranked plants into categories based on how frequently we think they occur on serpentine. So we called plants strict endemics if they occurred on serpentine in at least 95% of their populations. And then you go down the scale to plants that only occur on serpentine about half the time. And if they occurred on serpentine less than half the time, we didn't include them in this project because we figured that's not a very strong association. And so the exciting news that we have to share with you is that the proportion of serpentine endemics of the California endemic flora has increased quite a bit since the original estimate in 2005 with the taxonomic revisions and newly described species. It's now, we now believe it's 14.7% of the California endemic flora, which is really huge when you think about how serpentine is just 2% of California's area. But one reason for this increase is also because the total number of California endemic plants, actually the estimate decreased a little bit since the original study, so we had changes in both the numerator and the denominator for this figure. But there were a number of newly described serpentine species since 2005. So these are all species that were just described in a few years, between 2015 and 2018, I believe. So we had an Antenaria, a couple Monardellas, a couple Navarretias, a Brodea, and a few Sedums. But in addition to adding new species to the list, we did remove some species that had been on the list before. In some cases, species were removed because of taxonomic changes that lumped them in with something else that wasn't strongly associated with serpentine. In other cases, it appears we don't really know how things got on the list in the first place, but they shouldn't have ever been there. Calicortus greenii has never been found on serpentine, but it was ranked as a strict endemic in the original list. As all of us community ecologists know, you can't trust any species list completely. So hopefully no one did anything too pivotal using that original list. It was mostly right, though. We found very few mistakes, when you consider the length of the list. And so the rank order of the main genera and families for serpentine endemics was shaken up a little by the taxonomic revisions. Eriogonum and Streptanthus are now the two most serpentine endemic rich genera in California. And the biggest families for serpentine endemics are Asteraceae, Brassicaceae, and Polygonaceae. So a couple big shakeups here were Liliaceae used to be number 3, but because of splits, it was pushed way down the list. And Scropulariaceae used to be number 5. And of course, now it consists solely of like Mullen and Scropularia. No one really knows what's in Scropulariaceae anymore. But it doesn't rank very highly on the list. And then when we divide the taxa up by growth form, you can see that the majority of the plants are perennial forbs. A lot of perennial forbs, which, when you think about a serpentine plant that comes to mind, good chance it's a perennial forb. We also have a lot of annual forbs, a good diversity of shrubs as well, and then very few perennial graminoids, trees, and I think just one annual graminoid. So yeah, no big surprises there, and this was not a big change from what the original study had found. And geographically, no big surprises either. Most of the serpentine endemics are in the Klamath Range and the North Coast Range. The legend got left off of this figure accidentally, but the lower dark part of the bars is species or taxa that only occur in that region and nowhere else, and the light part of the bars is taxa that occur in multiple regions. So the two northernmost regions in the state contain more serpentine endemics than the rest of the state put together, which is what we already knew before these revisions. No big shakeup. So I know what you're thinking now. You're wondering, how can I access this highly informative and useful list for my own nefarious purposes? And I'm happy to share with you that it's really easy to do that. There are a couple different ways you can get this list. So our friends at CalFlora have updated the ultramafic affinity ranking based on our research. So if you go to a species page on CalFlora, down in the lower right-hand corner, there's that little ultramafic affinity ranking. And I have to say, I'm really impressed by CalFlora. They had that on the website within 2 hours of the time I emailed them the updated list. I was thinking like maybe in a year or something we would see it on there. It was absolutely shocking. So whoever made that happen at CalFlora, we are really impressed. Thank you. But if going through species by species on CalFlora is not going to satiate your thirst for serpentine affinity rankings, you can also download the entire list on my website. I put the link to the publications page there, and I'll put a QR code up on the next slide. So here's a link to the paper. We published this paper in Madronio in 2020. So all these figures I showed, or most of these figures, they wouldn't let us put the word clouds in. Harsh editorial standards. No, just kidding. But you can download the paper, and you can also download the spreadsheet that lists all the species and their rankings on that other list. And I just want to say we're really grateful to everyone who made this project possible. It was a long process that involved consulting with a whole bunch of people from all over the state. Way too many botanists to list them all on this slide, but thanks to everyone, and I know several of you are here in the audience. And we're also grateful to our student Micah, who helped out with a lot of the database management work. So that's everything I have. I think that was a pretty short talk, so we should have plenty of time for questions. Thanks.
Every 5 years or so. Oh, thank you. Oh, okay, a little better. I was curious to know, since this is all digital now, if you had it in mind to make maybe every 5 or so years, make little tweaks and updates and issue revisions.
Yeah, that's a really good question. That's something that's been on my mind too, because of course, as soon as you publish the list, you start finding things that need to be updated almost immediately. Yeah, I think that's a good idea, and I've been thinking about what the best way to go about that is. I was considering maybe setting up a website where we could have kind of a living list, kind of like the Jepson eFlora, and have a forum where people can submit suggestions, because it really would be great to have everyone's input, since so many of us are working in some little corner of California where there aren't too many people botanizing. So yeah, I'd be curious to hear if anyone else has suggestions or thoughts on how we could develop this into a living list. But yeah, I think that would be a great thing to do, and that's something I'll talk to Hugh about.
Any other questions?
Thanks, Jesse. I just wondered if you thought about doing a version of this that would be based on the California floristic province instead of the state of California. It'd be interesting to see how that plays out. And as you know, there is a species list for the CFP that denotes things as endemic or not. So just saying.
Yeah, I like that idea. So I'm thinking about, how it would actually change if we did that. Because there's not really serpentine in the Great Basin part of California. So it would really just be the additions of the Oregon stuff and maybe Baja stuff, right? Yes, the denominator, yes. Okay, interesting. Yeah, I always didn't love that we were using political boundaries rather than ecological boundaries for this. So, yeah, that's something I would like to see. Maybe we can collaborate.
I know you're a Southern Oregon guy.
Yes, I'm a Southern Oregonian in my heart, absolutely. And it's a great injustice that they've been walled out of California. Yeah. So, we got one.
So my hunch is that a lot of our serpentine endemics actually are just wimpy competitors and maybe would be less endemic to serpentine, or they're limited to serpentine as a reflection of what's going on everywhere else. And what's going on everywhere else has changed a whole lot over time. And so I wonder if either you have any hunches about, have any of these plants become more endemic to serpentine because the scene in grasslands is so challenging these days? Or do you think about how this changes through time, or is there capacity to look at that as herbarium records maybe capture some of that older situation?
Okay, that's a really interesting question. So yeah, whether I guess everyone probably heard that, whether serpentine endemism itself might be changing. So it sounds like you're suggesting maybe because of exotic species and grasslands outcompeting them, if things could be more and more relegated to serpentine. I mean, I guess I'm curious how much exotic grasses actually are increasing in the modern era. I don't know the answer. I mean, I know a lot of these exotic grasses have been here for a long time, but are they? Okay, let's do a poll. How many people here, and this will be legally binding, how many people here think exotic grasses have substantially expanded their cover in California in the last 10 years? Okay, okay, how many people think they have not changed that much in the last 10 years? Wow, no one. Okay, so that settles it. I think serpentine endemism is increasing. Susan, you look like you have something to say about this.
Kind of related to your question, related to your question, except not related to the invasive grass part. Hugh Safford had a really interesting speculation. Why are there so many fewer serpentine endemics in the Sierras than in the Coast Ranges? Maybe it's because there's more granite. Right? Because a lot of these plants just like rocky outcrops, and in the Coast Range, most of the rocky outcrops are serpentine, but not in the Sierras. As far as the invasive grasses, well, Marina here is really the expert, but it's my sense that when things get confined to serpentine, mainly because of exotic grass invasion, they don't end up as endemics. They just end up as things that are kind of hard to find, but not impossible to find off of serpentine. But you should really talk to the serpentine invasive grass interaction person about that.
Yeah, Marina, why don't you weigh in?
Yeah, I think grasses are driving a lot of natives to only being able to survive on serpentine. But as Susan pointed out, yeah, if they're endemic, then you're just not. Sorry. If the species are endemic to serpentine, then you're not going to find them anywhere else. But I do think grasses are really affecting natives' ability to survive in those non-serpentine areas.
Okay, we have time for one more question if anyone else has one. Just taking a picture of this beautiful super spreader event here.
How much of a threat are things like edgelops, which actually are adapted to serpentine and are starting to compete with some of our serpentine endemics?
How?
Goat grass.
Oh, egalops. Yeah. We all say the names differently. I don't know. What do you know, Marina?
I don't know. So it occurs mostly on serpentine soils, right? Yeah, I'm not sure. Are there any goat grass experts here?
Yeah. Who's the goat grass people?
Yeah.
You know they're here.
In my talk, which will be coming up in a few, I think I'm third, I'm going to be talking a little bit about some work showing how goat grass has affected serpentine through declines in forbs, and then I'll be examining how restoration of a serpentine meadow through removing goat grass has affected plant-pollinator mutualisms, and it has some pretty big effects, so stay tuned.
Awesome. Stick around.
We planted that question to entice you all to stay for the rest of the symposium. Okay, it's 10:20, perfect timing. So I'm really happy to introduce our next speaker, Michael Mulroy. Michael is finishing up his master's at Cal Poly, the original Cal Poly, not the new fake one. Just kidding. All respect for Humboldt. Big fan. And Michael is a really well-rounded naturalist. He's great with plants. He's great with lichens, which he'll be talking about today. And he's also a great birder. So the kind of person that's really fun to go out in the field with. And he's going to talk today about a really cool project and a really ambitious project analyzing serpentine lichens. And the title is Serpentine Lichens, Regional and Local Patterns of Assembly and the Challenge of Context. So welcome, Michael.
All right. Thanks, everyone, for being here. I'm really excited to give this talk, and thanks, Jesse, for the introduction. A little spoiler, serpentine lichens aren't maybe not quite as fascinating as the vascular plants on serpentine, but they're still really cool, so stick around. And I just wanted to mention I'm a member of the Geoecology Lab, which is headed by Dr. Nishi Rajakaruna. Just throw that out there. And so, yeah, what I want to talk about, mainly I want to get to my sampling project where I compare lichen biotas of serpentine and sandstone along a maritime gradient. So I'm hoping to get to that and have plenty of time for that. But first I want to set the stage with some background. First on what lichens actually are. So traditionally and probably most usefully, a lichen is considered this combination of a mycobiont or fungus and a photobiont, which is usually always some kind of photosynthesizing organism like a green algae or a cyanobacterium, as a couple of examples. So the photobiont provides sugar via photosynthesis to itself and also to the microbiont component. The microbiome provides this really nice controlled environment for that photobiont to grow and thrive in, protects it from things like desiccation, UV exposure, etc. So some useful terms for lichens are three main growth forms. So crustose growth forms are these really tightly oppressed lichens that are pretty much unremovable from their substrates without taking some substrate with it. Foliose lichens have an upper and lower surface but are still kind of growing along their substrates. And then fruticose, which I would argue are the most charismatic lichens, not the lichens I'm really dealing with in my study, unfortunately, but they're these more plant-like, shrubby types of growth forms that are more three-dimensional. So lichens can grow on pretty much anything if you give them enough time. That includes anthropogenic substrates and obviously natural substrates. And I just want to point out a few sort of important characteristics of substrates that affect what types of lichens can grow on them. The pH surface environment of a substrate is really important. Its water holding capacity, its elemental composition, these are all things that are shown to influence the community composition of a given substrate, as well as plenty of other factors as well. So why should we care about lichens of serpentine specifically? So for me, the main reason is that they're a really poorly documented yet major component of biodiversity on serpentine outcrops. This is very speculative, but I would speculate that in a lot of cases, the number of taxa of lichens on a given serpentine rock outcrop environment outnumber the number of vascular plants in a lot of cases. They play really ecologically significant roles in general, including in these serpentine outcrop environments. So they play roles in nutrient cycling, soil formation, and they probably have significant relationships in vascular plant succession and other ecological relationships with plant communities, although this hasn't really been, as far as I can tell, very well studied. Although it has been documented in other types of rock outcrops. Serpentine may harbor regionally unique and rare taxa, as well as unique lichen biotas, and they're definitely beautiful. They add a lot of color and texture to the landscape, which I think is sort of an underrated aspect of them. And a lot of people sort of see rocks and they don't really think about what is causing the colors that you see, but lichens are definitely playing a role there to the aesthetics of our world that we live in. So before I get into my sampling study, I want to set the stage. My co-authors and I published a Lichens of Serpentine Substrates review in North America in the journal Botany earlier this year. And so what we did is we aggregated reports of serpentine lichens from the published North American literature, and we classified every lichen taxon reported from the literature that we could find in terms of its substrate pH affinity, as well as its substrate type affinity, so what it prefers to grow on. From there, we tried to assess and analyze patterns of serpentine lichen communities, specifically in North America. And I'll just add, serpentine is probably the most well-studied rock substrate for lichens that exists. So there are some really good worldwide, global scale reviews on this topic as well that I would encourage you to check out if you're interested. But yeah, this was kind of honing in on North America. So overall, we found 476 taxa reported from serpentine in the published literature. Most of these are acidophytic, so they are low pH, kind of acidic pH preferring lichens. Really interestingly, there's always seems to be a consistent number of high pH preferring lichens that are co-occurring in these serpentine environments. Taxa with disjunct populations is really common on serpentine. Probably most interesting for this audience, there's very little evidence, basically no evidence of strict serpentine endemism, which makes me a little sad. I want to be able to say there's a lot of cool serpentine endemic lichens. That's just not the case. I think worldwide there may be one good case to be made for a lichen that's a strict serpentine endemic, and that's until you find it on some other rock type, which is always TBD with these lichens because they're not really well surveyed. And we see large site-to-site differences on small scales, as well as evidence for regionally unique lichen biotas. All right, so getting to my sampling study here. I'm doing a comparison of serpentine and sandstone lichen communities along about a 70-kilometer maritime gradient. And so what I'm interested in is seeing how community composition changes in response to a few different abiotic factors. And so the three groups of abiotic factors I was interested in are differences in substrate, so serpentine versus sandstone, climate factors related to maritime influence, so how close you are from the coast and sort of what effect that has, as well as some aspects of microtopography. And the main one I looked at and we'll talk about today is the effect of rock aspect on this. This is sort of an overview of my experimental design. I sampled 16 rock outcrops in total in and around Slope County. 8 of these were serpentine and 8 of these were sandstone. Some were coastal, some were sort of in intermediate distance from the coast, and some were inland. And at each site, I sampled 8 quadrats. It doesn't sound like a lot, but when you spend an hour in front of a 20 by 20 centimeter quadrat and you're still finding new lichens, it starts to sound more reasonable. So at each quadrat, I identified individual morphospecies. I took some abiotic variable measurements, and at each site, we took rock samples that we analyzed for elemental chemistry. Then we did voucher specimen ID for each morphospecies identified. We collected a voucher specimen and took, countless hours trying to ID these things in lab. Lichen ID is very hard and time-consuming. And I still find that to be the case today, and I've had a lot of help, which is the only way I could have done this project. So this is a larger sort of view of our study area. And yeah, I think I've said it all there. Here's a couple of examples of sort of the sub-outcrops within larger sampling sites, and our quadrats are placed there. So overall, again, just interested in understanding how community composition responds to differences in abiotic factors. We had a couple of hypotheses, neither of which are supported by my data. That's a little spoiler alert. My first hypothesis was that we'd have higher richness along the coast, less richness further inland. And my second hypothesis, which I thought was really cool, and I was kind of bummed that it wasn't the case, we thought we'd have greater similarity between serpentine and sandstone communities along the coast, and then more differentiation between those two substrates further inland. And our data definitely does not support that. It might be the opposite, if anything, but probably not even that. So, oh well. So here's just some pretty representative quadrats from my study. These are sandstone sites. The south-facing aspects are on the north, or on the top. North-facing on the bottom, just to give you a sense of what we're staring at for hours when we're doing this field work. These are serpentine sites, and one thing I'll just point out is the inland, south-facing serpentine sites is pretty typical to see really bare-looking quadrats. We never had a quadrat, and these were randomly placed. We never had a quadrat where we didn't actually find any lichens. We always found something in some little crack somewhere, but they were looking pretty bare. Okay, quick summary of my results. Overall, in my 128 quadrats sampled, we found 135 lichen taxa. Most of these were chlorolichens, but we also had 9 cyanolichens, which are lichens that use cyanobacteria as their photobionts. I'll talk a little bit more about that in a minute. From serpentine, we had 80 distinct taxa. From sandstone, we had 100, and then we had 45 taxa that were shared between the two, that occurred on both substrates. 22 new records for lichens from serpentine in North America. So that's 22 additions to that list, which I'm pretty excited about. In terms of richness and diversity, again, we didn't have that sort of higher richness along the coast that I thought we might have. And the main pattern that stuck out was that sandstone sites and also quadrats had higher species richness. This was significant for sites, not statistically significant for quadrats. And then the other pattern that I just kind of found interesting, but I have no idea how to explain, is that coastal sites, the paired sites, we sort of sampled sites in pairs. I forgot to mention that. Our paired sites on the more coastal sites had higher richness on sandstone, and then that trend reversed itself on the more inland sites where there was higher quadrat level richness on serpentine, which I just think is interesting. There's a lot going on in this figure, but the main takeaway is in this ordination plot on the left there. You can just see that there's a pretty good separation between serpentine, which are the blue dots, and sandstone, which are the tan dots. And so the community composition between these two substrates, on average, is pretty different. And then a couple of other nice clean patterns that I just want to mostly finish up with are species richness being higher on north aspects of rocks, and this is true both on sandstone and serpentine. And it's also seems to be the case throughout our maritime gradient, which is what that right-hand graph is trying to show there. And a similar thing going on with percent cover of lichens. There wasn't a statistically significant difference for sandstone, but a very significant difference between northern and southern aspects for lichen percent cover, with those northern, less sun-exposed aspects having much higher lichen cover. And then the disparity between north and south appears to increase quite a bit as you go further inland. So, those south-facing rock outcrops, or those south-facing aspects on inland serpentine sites that are just baking in the sun have just very low lichen cover. We also had a few serpentine-associated lichens. We did indicator species analysis. So these are our 7 most strong serpentine-associated lichens. These either exclusively occurred on serpentine in our study or just occur on them most of the time, with very few occurrences on sandstone. So three of these are in the genus Polytechnic. Those are the brightly colored ones. We also had 3 cyanolichens that were strongly associated with serpentine, and this one Phalloidema ion, which is either an obligate or facultative parasite when young on cyanolichens. So that kind of explains why it's occurring mostly on serpentine, or actually exclusively on serpentine in this study. And then, just to mention a little bit more, there's a really strong cyanolichen-serpentine relationship where 8 of our 9 cyanolichen species were either only found on serpentine or strongly associated with serpentine, and I have no idea why this is the case. And I'm curious to kind of delve into this. I haven't found anything in the literature that sort of suggests or has talked about this. All right, so take-homes. My main take-home from this is just that there's very high diversity of saxicolous lichens, rock-dwelling lichens along the central California coast. Again, we had 128 20 by 20 centimeter quadrats that we sampled, and we found 135 lichen species in these quadrats. And so that's a fraction of the true species richness in these rock outcrop environments that we looked at. Rock type and climate definitely influence community composition. We have that cyanolichen affinity for serpentine that I talked about. And then that really nice clean difference between north and south-facing rock aspects for both serpentine and sandstone. And then also that many lichens do have a clear substrate preferences, I think is another clear takeaway. But what I really want to emphasize is that these results are interesting, but they're also really hard or challenging to put into context because there just aren't a lot of studies of rock-dwelling lichen communities out there. And serpentine is actually one of the most well-studied substrates for lichens. So we don't have sort of comparable amounts of studies looking at lichens of limestone, granite, etc. So I'm really excited to be able to compare this data with more studies that are similar. Here's some interesting finds and range extensions that I think I probably don't have time for, but if anyone wants to talk about some fun range extensions and oddball lichen finds that we have for this study, I'd be excited to talk to you about them some other time. And a big thank you to the California Native Plant Society SLOW chapter. They helped fund this study. And then a special thank you to Jason Dart, who helped me just a ton with the lichen microscopy and also with my field work. He couldn't be here today, but thanks, Jason. I know you're out there somewhere. And with that, I'll take any questions. I don't know how much time I have left.
I think we have 5 minutes for questions.
Hi, really cool study. Lichens are awesome. Did you say that you saw a lot more cyanolichens in the serpentine habitats? So are you thinking that's contributing to the higher diversity you saw in those habitats possibly? Like there's just lichens, cyanolichens, and, other ones. I'm not thinking of the name.
Yeah. Chlorolichens is the other sort of main type that we found. They contribute a little bit, but there were only 9 cyanolichen species we found in our study, so they were definitely in the minority. But yeah, most of them were strongly associated with serpentine. We had one that only occurred on sandstone, and it was in one single quadrat that we found it on. So yeah, I think that is a possibility, and it's also possible that there's like a relationship with like cyanolichen occurrence and occurrence of other species. Like there is that kind of obligate facultative or obligate parasite that like needs cyanolichens to occur itself in any given place. So yeah, thanks.
So I have a question about elevation. Did you note the elevation of your findings, and were there any influences regarding the elevation?
I can speak to that anecdotally because we did have some sites that were close to about 1,000 meters elevation, but all of our sites were under 1,000 meters elevation. So that was sort of something we tried to sort of control for to the extent that we could on like a coast inland gradient where you're going to have some amount of elevational change. But on some of our sites, we did find some species, like a few species that are more characteristic of montane environments. So they were probably at the lower ends of their elevational range. So yeah, that's something we did think about a little bit, yeah.
We have another question back here.
So on your south versus north facing slope issue, how long does it take lichens to establish even a small little dot, let alone a gorgeous patch like that? On the south facing slopes in the area where I work, which is a lot of sandstone and a lot of serpentine, you just have a lot of exfoliation of the rocks. And so how does that tie in?
Yeah, so that was one of the substrate factors that are important to lichens that I actually wanted to mention. And another variable that I didn't mention in this talk that I think is relevant is for our sandstone sites, we see like in some sites there's pretty clear exfoliation, which I think influences the community quite a bit. And we also have some really hard sandstones. And this wasn't a variable that we were initially planning on looking at, but I would like to look at it. We have a geologist that we work with who's sort of going to look at the differences in mineralogy and cements between our sandstones to sort of get at that question because it's super relevant for lichens, absolutely.
I think I saw your hand last one.
Hi, Michael. Great talk. So cyanolichens have cyanobacteria, which can fix nitrogen, right? So do you think there's something potentially going on there with like serpentine being generally a nitrogen-poor substrate and these nitrogen fixers being more abundant there?
That is definitely one train of thought. There are a few others, but that has been like suggested. And yeah, so potentially. Yeah, I'm not really sure to the extent to which nitrogen is a limiting nutrient in these communities, especially since we're not super high elevation. So like I went to the nitrogen deposition talk, and some of our sites are close to 1,000 meters, but and we're not in the high nitrogen deposition area. But I kind of wonder about how limiting nitrogen is in these environments. But yeah, it's a great question. That's something I'm going to consider for sure.
Hi. Would you go over quickly your collection methodology for the really crustos stuff, like for the voucher specimens?
Sure, yeah. The methodology was like doing the best we could because these crusts are just insanely hard, and so many cases where you think you got the morphospecies and you made even multiple collections, and you have two things that under a hand lens look pretty much the same, or at least within the taxonomic variability you'd see in the species, and they're different species, or even like different genera. So yeah, it's just a challenge. We did site revisits to sort of try to adjust that, but there's some inherent, squishiness in this data because, it's just hard to really get good IDs in the field for some of these things, for sure.
All right. Our next speaker is Becca Nelson. Becca is pursuing a PhD in Dr. Susan Harrison's lab at UC Davis and is co-advised by Fernanda Valdovinos. Becca researches how invasive species and restoration strategies affect serpentine plant-pollinator mutualisms in California grasslands, and she's broadly interested in global change and community ecology. Welcome, Becca.
Thank you, Marina. Hi, everyone. Good morning. Today I'm going to be talking about the effects of invasive species on serpentine plant-pollinator mutualisms. So before I dive into my work, I'd just like to acknowledge all of the folks who have supported me in this research, particularly my advisors, Susan and Fernanda, as well as additional collaborators, Paul Agner and Sabine Dritz, and all of Paul and Kathy's support at the McLaughlin Reserve. Thank you. So we've talked a little bit today about how serpentine prairies are these refugia for California's native plant and pollinator diversity. They also support quite a diverse community of pollinators and insects as well. And despite them being these important refugia from things like California's more invaded non-serpentine grassland, there's still ways in which invasive species can dramatically affect serpentine grasslands as well. There's sort of two primary mechanisms that I'm exploring in how these invasive species affect serpentine. One I sort of hinted at a little bit earlier. The serpentine-tolerant invasive species, particularly barbed goat grass or Aegilops triuncellus, can have direct effects on serpentine plant communities by forming dense monocultures that decrease the diversity and abundance of native wildflowers in serpentine. Additionally, serpentine can often occur in these very patchy mosaics with non-serpentine habitat, and so invasive species such as hairy vetch that occur in the non-serpentine can share pollinators with serpentine native flora, and there can be potential for some indirect effects from these non-serpentine neighboring invasive plants on the pollination and fitness of serpentine plants through the movement of these shared pollinators between serpentine and non-serpentine habitat patches. So I'm going to share some research that I've done exploring these two pathways, starting with sort of the effects of goat grass as a serpentine-tolerant invasive species. So I asked, how does removing goat grass as a restoration of practice affect pollinators and plant pollinator communities in serpentine prairies? So to begin with, I'm going to talk a little bit about some hypotheses I have that motivate this research. So I think that removing the goat grass can potentially be beneficial for pollinators through two ways. One is because it forms these dense monocultures. By getting rid of the goat grass, you may be opening up more niche space for serpentine wildflowers that are a much better floral reward than the wind-pollinated goat grass is for serpentine pollinators. Additionally, because goat grass forms such dense patches, it can cover the ground. So by getting rid of the goat grass, you may be opening up more patches of bare ground that create a better quality of nesting habitat for ground nesting bees. So I'm predicting that these can result in boosts to increased pollinator abundance and diversity when removing the goat grass, and that this can further have changes in the architecture of plant-pollinator interaction structure within these serpentine prairies. So for this study, I leveraged an existing restoration experiment that my collaborator Paul Agner established at the UC McLaughlin Reserve in the Coast Range, where he selectively removed goat grass through a variety of methods within a serpentine meadow, and then compared those to serpentine plots where goat grass was retained as a control. Paul and his interns then collected 3 years of pollinator observations on these plots, which I then analyzed using a network approach. So to dive into some results, to start with, these are showing sort of responses of the plant community to goat grass removal, and the asterisks denote significance. So we found that the goat grass restoration practices were effective in significantly decreasing the cover of goat grass in these stored plots relative to the control. And likewise, although goat grass was our main exotic annual, in general we found that there was a significant decrease in exotic annual grasses in response to these restoration practices. There was likewise an increase in wildflower abundance, particularly for all preferred wildflower species that were visited by pollinators in our pollinator data, which you can see those wildflowers in panel D. And then also there was a significant increase in the goat grass removal plots for our most abundant native wildflower species at the site, which was Lichens californica, also known as goldfields. We then investigated whether these changes in the plant community connected to changes in pollinators, and we're happy to find that in the restored plots where goat grass had been removed, there was a significant increase in pollinator diversity in terms of richness, pollinator abundance, as well as pollinator Shannon diversity relative to control where goat grass was retained. There was also a significant increase in the abundance of ground nesting bees in these restored plots relative to the invaded control plots, although the overall proportion of ground nesting bees relative to the whole bee community did not significantly differ between the control and restored plots. So these results suggest that removing goat grass as an invasive species can increase the abundance and diversity of pollinators. This can then result in changes to the overall network of plant-pollinator interactions as well. So the way to break this diagram down is that the top row, each of those little rectangles represents a different pollinator species we observed in the data set, whereas each of the bottom rectangles represents a plant species, and the links between them represent an interaction observed between that plant and pollinator, and the thicker the line, the more frequent that interaction was observed in our network data. And the red rectangle represents the goat grass, which only appears in the control network, and although some insects landed on it, since it's a wind-pollinated grass, it's unlikely they were really receiving much in the way of floral rewards from it. The yellow box with the little picture is the goldfields, or Lichens californica. So we found that these goldfields acted as a critical hub for pollinators in the network, attracting a diverse community of pollinators that spanned all of the pollinator functional groups we identified at our site, and that with restoration, the relative influence of these goldfields in affecting the architecture of the species interaction significantly increased. And there was likewise an increase in pollinator niche overlap in these restored plots, with a greater amount of and diverse amount of pollinators visiting the goldfields. And so because we have a lot more types of pollinators visiting the goldfields in this restored network, that sort of increases the functional redundancy of pollinators in the network, which can be a stabilizing factor. We also looked at something called network nestedness, which is sort of a way of describing how interactions within the network are subsetted like Russian nesting dolls. So in a highly nested network, you get generalist plants or pollinators that interact with both other generalist and specialist species, but then your specialists in the network only are reliant on interacting with other generalists. And interestingly, we found that with removing goat grass as a restoration practice, the nestedness of the network significantly decreased, which in some ways contrasts with previous studies that have found that in more biodiverse networks, they tend to have higher nestedness. So this is something that I hope to explore further, sort of what might be driving this in future work. So some takeaways from this first section are that removing goat grass, thanks to all of Kathy and Paul's wonderful efforts at McLaughlin, was a very effective restoration strategy for serpentine prairies, increasing not only plant abundance of wildflowers, but also pollinator abundance and diversity, and that these changes through restoration in serpentine resulted in also changes in the architecture of plant-pollinator interaction networks, increasing niche overlap and functional redundancy, but also decreasing nestedness. And from a management standpoint, Lichens californica, or goldfields, played a really core role in the network as a major hub, suggesting that sort of establishing some of plants sort of like Lichens for restoration can make a big move towards really improving pollinator services in restoration efforts. So we talked a little bit about invasive species within the serpentine, but we also know that serpentine is often contained in a matrix of very invaded non-serpentine grasslands. So now I'm going to pivot to talking a little bit more about the second pathway, about how hairy vetch, an invasive legume with a very attractive floral reward for pollinators, affects potentially the pollination of serpentine native plants through the movement of pollinators across an ecological boundary. So I ask, how does proximity to vetch at these serpentine, non-serpentine boundaries affect plant-pollinator interactions and pollination within serpentine prairies? So what motivates this research are the idea of cross-boundary effects, which is when components in one habitat have effects on components in another habitat. So traditionally in ecology, these boundary effects are often studied in interfaces between aquatic and terrestrial ecosystems, where you might get seaweed that washes up on a beach, and then that seaweed provides nutrients for a terrestrial island food web. So a recent review pointed out that these potential for boundary effects affecting ecosystem function and diversity have been pretty understudied in boundaries between terrestrial ecosystems, as well as for mutualistic interactions like plant-pollinator relationships. So my research aims to address this gap by examining how do pollinators that visit the invasive vetch in non-serpentine patches, do they have potential boundary effects on native plants such as this bull clover or Trifolium fucatum in the serpentine through moving across this serpentine, non-serpentine boundary? And in particular, these two plants are visited by highly mobile pollinators such as honeybees and bumblebees that tend to move across the landscape. To test this question, I collected some initial data last spring at the McLaughlin Reserve by observing patches of bull clover at varying distances within a meadow from where the patches of invasive non-serpentine vetch were, but then also had several of these boundary meadows at a landscape scale that spanned an invasion gradient of vetch based off differing levels of vetch abundance. And so in addition to focusing on pollinators to this focal species, a native legume of bull clover, I also recorded plant pollinator visitation data for these serpentine meadows at the community scale as well. I'm now going to walk you through some very preliminary findings that I have for this section of my research. So within meadows, the x-axis here shows sort of distance within meters from vetch, and then the y-axis shows pollinator richness and pollinator abundance on the native bull clover. And I found a slight increase with distance away from the vetch in the pollinator richness and abundance. However, this effect was not statistically significant, sort of pointing to the need for further exploration on this. At the landscape scale, we see something a little different. So this is now showing sort of invasion intensity level of vetch, either low, medium, and high, for all these meadows at a more landscape scale. And the letters on the graphs denote pairwise differences in significance. So here I find that there's sort of this peak abundance of pollinators on the bull clover, as well as peak diversity of pollinator richness on bull clover at intermediate invasion levels, which is consistent with some prior work that has found that sometimes at moderate intensities of invasion, invasive species can sometimes potentially have facilitative effects on native species by bringing more pollinators overall into the area. And here too in the networks we can see that there's sort of this change in structure with invasion level sort of shifting towards vetch being more dominant in the network, and at high invasion, vetch replaces the bull clover as being a core hub in the network. So this is something that I'd like to explore further by sort of doing a more nuanced spatial analysis that integrates these scales. Thank you, everyone. If you have questions or would like a preprint from the Goat Grass project, please don't hesitate to reach out. And you can also check out my website with this QR code to learn more about what I'm working on. Thank you, everyone.
Do you have a listing of the other native bees that you found on the site, or can you mention a few of those?
Yeah, that's a wonderful question. Thank you. So one really cool finding that we had is that we have 4 different species of native bumblebees at our site that are like among the primary pollinators for these legumes. Yellow-faced bumblebee, California bumblebee, and also black-tailed bumblebee. But our 4th native bumblebee that we found was actually a really exciting surprise for the McLaughlin Reserve, as it's the Crotch's bumblebee, or Bombus crotchi, which is an endangered, it has a very funny name. It's named after some old-timey entomologist whose last name just happened to be Crotch. But yeah, it is a cool bumblebee that has, it's endangered, and it's in the process of being listed endangered not only at the IUCN but at the state level. So I'm also been collaborating with the reserve to sort of document this endangered bumblebee at our site. And there's also a lot of other cool bees like mason bees, andrenid bees, sweat bees as well. Thank you.
Hi, is this on? Okay. What's the time frame between? Hold it closer. What's the time frame between when the goat grass was removed and you did the pollinator study?
Yeah, that's a great question. So the goat grass removal, I think, started sort of in the late 2000s and then was sort of wrapped up around 2011. And then once Paul wrapped up sort of the phase of these removal treatments from about 2011 to 2013, Paul and his interns then collected floral visitation data on them. And sort of I did also, one result I didn't show was that I looked at whether over the time since restoration for those 3 years, if there was differences in pollinator composition sort of within the meadow, and the strongest differences were within sort of that first year of restoration, but the overall boosts in richness were sort of retained throughout all 3 years of the data collection following the restoration efforts. Thank you. That's a good question.
Any other questions?
I'll be a repeat offender. I was curious to know if there was any information regarding the relative effectiveness of the different treatment types for removing the goat grass. You mentioned, like, I think 3 or 4 different methods at the beginning. And if you could just speak to that, what was more effective, what was easier?
Yeah, that's a great question. So the treatments that Paul and Kathy used were grass-specific herbicides targeting goat grass, hand pulling the vetch, as well as mowing as like mechanical means of removal. And from the perspective of pollinators, we did not find any significant differences in whether any treatment was more effective in increasing pollinators. They were all effective at removing the goat grass as well as increasing pollinator diversity and abundance. So from a management perspective, I think potentially any of the treatments can be effective, and it can kind of depend on like resources and what might work best for a given reserve. I think one thing that Kathy's sort of highlighted a lot is that there sort of does need to be sort of this sustained removal of the goat grass. So although these meadows at McLaughlin are restored and sort of much lower in terms of their goat grass, it's sort of this continual effort that Paul, Kathy, and a crew of interns put in every year to sort of use combinations of these methods to sort of every year go back and continue to sort of mitigate the reestablishment of goat grass in these systems.
All right, I think we have time for one more quick question. Is 11:01? All right.
Thank you. I have kind of a basic question, but how long have you been doing this research, and in what interval do you do your monitoring? And have you seen changes over time?
Thank you. Yeah, so I'm currently in my 3rd year of my PhD. So for the second part with the vetch, I have about 2 years of data on this research, and I'm hoping to collect a few more years. For the goat grass project, that data was collected from 2011 to 2013 by my collaborator Paul and his interns, and I then sort of curated and analyzed the data, but they're still sort of actively collecting data for like my second part of my research in some of the same areas that first study took place at the reserve. So I'm sort of been working on it for like a couple years now and hoping to continue to work in this system and hopefully be able to try to build a more long-term monitoring program for pollinators, because I think that's something that's really needed, particularly for serpentine systems, is understanding how some of these functional shifts in plant community over time on serpentine might translate into implications for the support of insect diversity. Thank you.
Great. Thanks, Becca. Our next speaker is Elise Elwood. Elise is a graduate student at UC Davis, and Elise's research focuses on understanding how seed and seedling traits affect resilience to drought and in grassland plant communities.
Hi. So I'm Elise. I'm a PhD student. Hi. My name is Elise, and I'm a PhD student at UC Davis, working with Jennifer Funk and Jennifer Grimmer, as well as a collaborator, Anu Eskalainen, who's at the University of Finland. And I'm really excited to be part of our Serpentine session today, and I'm going to be talking about seed traits as a way to think about how global change factors are influencing our serpentine communities. So as Jesse started us off with, a really great definition of serpentine soils and how very cool they are with super high levels of endemism and lots of native diversity. And as Becca was talking about, there's kind of a mosaic of habitat, particularly at our study site of McLaughlin, but also just in general in Serpentine. It's not happening completely as a closed-off island, but exists on a resource gradient where we have really harsh, low-resource patches up to like being next to non-serpentine, nutrient-rich invasive grass dominated communities. And so for my talk, I'm going to be referencing this as lush serpentine and harsh serpentine. And so the harsh serpentine being the really coarse soils that have really high native diversity, really low water holding capacity, and our lush serpentine are more alluvial soils that have, an intermediate amount of nutrient and kind of a mix between native and invasive species. For this talk, I'm not going to be talking about the non-serpentine, but I just wanted to throw it up there to remind us that lush serpentine is really still serpentine and is very low nutrient compared to what we think of as non-serpentine. So one way to think about our communities is, and community composition is to think about how traits shape the filters that we see. So thinking about the species that we find within the region, there are certain abiotic and biotic conditions at every location, whether it's competition or really high temperatures, that determine which species are able to thrive there, and that this ability can correlate with species traits. And so if we can figure out which traits are important and relate to which abiotic factors, we can use that in conservation and restoration. So we already know that nutrients matter and that water matters and that it can affect the whole community. So just threw up an example of like a lake eutrophication scenario, and there's a picture of the beautiful superbloom. With the changing filters, we get differences in the communities, and we know that in serpentine habitat, when you have combination of nutrients and water, our above ground community shifts, and we have a lot more invasive grasses as opposed to native annual forbs. And we are just beginning to understand how this relates to seed banks and diversity underneath the soil. So in this graphic I have, the blue is kind of representing the soil, and then the different species underneath the soil as the seeds, and then the above ground community. So seeds matter. This is my fun moment to just be like, wow, look at all this diversity, and this is I could only fit 9 of my species up there. I wanted to fit them all, but I couldn't. There's just so many different things that vary, and we see so much diversity in seed traits, but it's not something that is really widely studied. Most of the time we just look at seed mass, which is not really sufficient to characterize all the traits, and so my goal here is to connect traits to particular abiotic filters and responses. So as I was saying, seed banks can contain hidden diversity, so we know that in serpentine systems in particular that the above ground community, and if that is changing with resources, that has a big impact on what is in the seed bank. And we also know that under times of low water availability, our native forbs do better in the seed bank, that they increase in abundance relative to invasive annual grasses. So in my little diagram here, going back to my filters, you can see that, so we have some abiotic filters already in Serpentine. It's very rocky, very high temperatures, very harsh magnesium ratios. And so some species are able to get through those filters and survive either above ground or in the seed bank, and it might depend on the year and the conditions. And so in this study, I'm thinking about if we're changing the abiotic filters, what is happening? So if we add nutrients, if we change the water regime, then we expect to see changes in our community composition. And so some species, like the little blue flower there, may be able to thrive in an altered environment after these global change factors, whereas they were not able to in the historical conditions, and vice versa. So this brings me to my question. So does resource availability affect our community-weighted seed traits in a seed bank community? And I'm going to look at this in two different ways. So the first way is thinking about how do seed traits differ between habitat types. So here I'm thinking about the lush serpentine and the harsh serpentine. And then the second way is to look at a experimental manipulation where we have control plots, nutrient addition plots, water, nutrient, and water plots to see how the seed bank differs between those treatments. And I'll look for if there is an interaction. So quickly, methods. So I'm using plots where there is an above ground treatment for 5 years in the field. So for both the lush and the harsh serpentine plots, they were either control, had nutrient addition, water addition, or both. And then after 5 years, we went in and collected a sample of soil from each plot and grew it out in the greenhouse and noted every single stem that came up and what species they were. So we have a really good quantification of what is in the seed bank, which is relatively rare because it takes a long time and effort to do. And then I did many trips to McLaughlin to collect as many species as I could, and in particular the most common abundant species in both the above ground and below ground communities. And so in my study I have 71 species, and they were collected not from the experimental plots, I want to note, but just from serpentine soils at the site. So there's lots of seed traits that could affect seed germination and persistence. For today, I'm going to be focusing on seed mass, seed coat permeability, and the carbon to nitrogen ratio. And again, there's lots of mechanisms of how these traits can be important. We know that seed mass affects the soil depth within the soil. It's like the seed goes into the soil, and also affects seedling survival probability when they germinate. Seed coat permeability and the carbon to nitrogen ratio can affect the likelihood that a seed is predated upon while it's hanging out in the soil, because we think about seed banks as a way to last through time, and every year that a seed stays in the soil, there is a risk that it'll be predated upon by a fungi or a microbe or another edible. But the traits can help us predict that, and also germination cues. And I just wanted to recognize that we expect these things to change with changing nutrient and water availability. So for my results today, I'm going to be using a community-weighted mean of each trait, and so that is the value of the trait taking into account the relative abundance within the community, and it kind of shows us a trait trend or strategy that is working well in that community. So my first question, or getting to the first result of does the community weighted mean differ between our different community types, so between our lush and our harsh serpentine? And it does. So we see really significant differences for all three of these traits, which is pretty fun. The carbon to nitrogen ratio is much lower in the harsh serpentine. So that means that there is more nitrogen in the seeds that we find in the harsh serpentine, which is really interesting, given that that's the community that has much less nitrogen available. For seed mass, we have higher seed mass in our lush serpentine. That's kind of what we expect with grasses having larger seeds and seeds that are closer to the soil, and more invasive seeds we think about being larger seeds, heavier seeds that can germinate and compete. The seed coat permeability was the most interesting, and oh no, it got cut out. But you should be able to see the lush bar is really low, and so the harsh serpentine or more nutrient-poor soils have much higher seed coat permeability, and they take in water much faster. This was really surprising to me. I think about invasive species or species that are trying to be really competitive, of taking in water and germinating really quickly. And so I'm still kind of wrapping my head around this. I'm wondering if maybe the harsh serpentine soils have such a low water holding capacity that when they do get that water, it's a really good cue for them. Whereas in the lush soils, there's water more often, and so it's less of a good indication. So still thinking about that if you have ideas. Next, I looked at the treatments, and so this graph is kind of boring, but basically it didn't find any difference. No difference between nutrient addition, nutrient-water addition, or water addition for carbon and nitrogen or mass. I find a few differences for seed coat permeability, but they really show up in the interaction and not individually. So moving on to the interaction. So on the right here, I have the harsh serpentine, and on the left, there's the lush serpentine, although the axes are not the same. And you can see that in the harsh serpentine community, when water was added, whether it was just the water treatment or the water and nutrient treatment, we see this really big decrease in seed coat permeability, which is cool because it is mimicking what we saw in the difference between the harsh and the lush serpentine. Whereas when we looked at the other traits, we didn't see this kind of parallel trend. And it's also interesting that water matters a lot, but the nutrient level doesn't seem to have any effect at all. And that in the lush serpentine, where there already is some water, we didn't see any impact at all either. So my key takeaways are that for seed mass, the lush communities have higher seed mass compared to our harsh serpentine communities. Seed mass is the most studied trait, but I just wanted to point out that, at least for me so far, it is not the most interesting one. For the carbon to nitrogen content, we see a higher ratio for the lush serpentine communities, so that they have more carbon and less nitrogen compared with our harsh serpentine. And for seed coat permeability, we see that both the habitat type and the experimental treatment show that water is more likely to reduce the community's seed coat permeability. And so maybe that trait is somehow related to a filter that is influencing which species are able to thrive in harsh versus lush serpentine. With that, do quick acknowledgements. Really grateful for all of the support I get from the Grimmer and Funk Labs. I've had so many really wonderful undergraduate interns. Couldn't do it without them. And also, quick shout out to the lab managers, Ava and Sarah, who've been so helpful. All right, and I'm ready for questions.
I would ask you about, like, multiple question kind of how far down do you research the seed bank? How far down do you generally go? And how long does it last? And what's the effect of animals on the seed? Fools or insects bringing, like ants or whatever, bringing the seed down into the soil?
Yeah, so first question was how deep in the seed bank did we look? We did around 5 centimeters, which in the serpentine soil is actually a lot. Most of the time it's hard to get that deep because it's so rocky. We don't know exactly how long seed banks last. But we think that it varies by species, but that in serpentine systems it lasts many, many years as a way that they kind of hedge their bets against low water availability. And then the last part was asking about animal interactions. Yeah, so there isn't a ton known about animal predation in serpentine soils. From what I've read, it seems more likely that there are fungi or invertebrates that are eating the seeds, and in particular, in communities that have more water, soils with more water, they attract more predators, and so that can influence the predation ratio. Also, something that I haven't looked at yet, but I'm really excited to, is seed size, and that I know that a lot of predators are kind of size gape limited in which seeds they're able to eat, and so that could have an effect.
Hello. Okay. Hi. I don't know if I missed it, but what exactly was the water treatment? Like, when was it? How much water? Growing season only, or?
Good question. Yeah. So the water treatment was applied for 5 years, and it was an extension of the normal kind of growing season. So after the rain stopped for the year, it was continued for another, I think it was like 5 weeks. And because this study was done largely during a drought period, we can kind of think of the results as more like the water treatment was more of a control kind of year, and that the normal control plots were kind of more like drought plots.
One more question? Anyone?
So I'm just wondering, did you notice any difference between the species composition for invasive species in the harsher serpentine soils versus the more lush? And if there were invasives that thrived more in the harsh, which were they?
That's a good question. I don't know off the top of my head, although maybe someone else does. I know that the harsh serpentine that didn't have any extra water or extra nutrients has very low invasive content, but then when you add the nutrients and add the water, then you get a lot more invasives coming in, both above ground and in the seed bank. I don't know off the top of my head which species in particular.
In the lush serpentine, we see a lot of your typical annual invasive grasses like medusa head, avena, lolium. It almost looks like a non-serpentine grassland. Yeah. And then the harsh serpentine is much more typical.
Thanks, Marina.
All right. Thanks, Becca.
All right, it's my great pleasure to introduce our next speaker. Michelle Coppoletta is an ecologist with the U.S. Forest Service Regional Ecology Program. She's worked in ecosystems all over California, from the towering dunes of the coastal lands to the ancient forests of the Sierra Montane zone. Yes, and she knows it all. The secrets that the landscape holds, they will be coming out in this last talk. And her greatest regret is that she's been so busy doing ecology, she's never been able to be a haunted house coordinator. Welcome, Michelle.
I did not provide that bio, just so you know. Thank you so much for having me. It's been really fun for me to sit in on this session because I think it just really emphasizes how unique and diverse these serpentine ecosystems really are, and also how much we still have to learn about them. And one aspect that I'm going to be talking about today is one of these fundamental processes in California ecosystems, which is fire, but which is actually pretty surprisingly not well understood in a lot of our serpentine ecosystems. And I did this project with a number of collaborators, including Scott Stephens, who was the lead on this project and who was supposed to be giving this presentation today. So that's really unfortunate that you have me instead of him because he's a really dynamic speaker, and I'm very sorry that he's not here to present this information. But I'll do my best. So today I'm going to be talking about some of the serpentine that occurs on one of the forests that I work on, which is the Plumas National Forest. We're situated in northern California at the far end of the Sierra Nevada mountain range. And we have some of the most extensive serpentine in the state. We have over 56,000 acres of serpentine-derived soils, a lot of tons of rare and endemic plant species that occur in these ecosystems. And actually about 33% of all of the rare species on the Plumas National Forest are actually associated with serpentine soils. And over the last 23 years, we've seen 73% of these serpentine outcrops burn. So as managers, managing not only these rare species, but these serpentine ecosystems and plant communities, it's becoming increasingly important for us to understand how fire has operated in the past and how it is currently operating in these ecosystems. So all of us know that these serpentine soils give rise to this really just unique and diverse number of plant communities. These just represent 4 common plant communities on serpentine soils on the Plumas. In the top left, you have those kind of classic serpentine barrens, those really open rocky outcrops with very little vegetation. We also have pretty shrubby serpentine ecosystems. And then some of the 2 that I'm going to be concentrating my talk on today are some of these more productive forested serpentine systems that we have. So I'm going to be focusing on some of our forest types that occur on serpentine, and these are pretty much mixed conifer forests. They're dominated by 2 primary species, Jeffrey pine and incense cedar, and they have kind of that classic serpentine problem, high levels of iron and magnesium in the soils, low levels of a lot of essential nutrients, and they often occur in these kind of shallow soils with pretty low water holding capacity. So compared to some of these more productive forest types that we see, these serpentine systems tend to have pretty low productivity and pretty slow growth rates. We actually aged some of the seedling size trees, so less than DBH, so about this tall, and we found that they were somewhere between 30 and 100 years old. So we have really slow growth rates on some of these serpentine soils. And in pretty close proximity, we have these other mixed conifer forests that are adjacent on these much more well-developed forest soils, and they're dominated by a pretty wide diversity of conifer species, considering, 4 or 5 different species and some black oak. They occur on these soils that are relatively deep, they have high nutrients, and much higher water holding capacity. So as a result, you see these much higher productivity rates and a lot higher growth rates on these systems. And what's really interesting, and I liked in one of the last talks you were talking about this kind of boundary between these 2 systems, but these 2 forest types are often found in pretty close proximity. So this is one of our study sites, and you can see there on the right the serpentine kind of grading into that non-serpentine forest type. And you can see that in the serpentine we have this kind of more open stand dominated by this kind of patchy vegetation structure. And as you move into that kind of non-serpentine, you're getting much denser forest, much higher cover. And so as ecologists, we start to think about, okay, we have these pretty distinct differences across this ecotone, differences in productivity and species composition and structure and biomass. How are ecological processes differing across this kind of gradient of productivity? And as fire ecologists who are living in and breathing smoke in the Sierra Nevada, we're often thinking about the role that fire plays and how fire might play a really different role in these kind of distinct but adjacent ecosystems. And surprisingly, the role of fire in Sierra Nevada serpentine communities is not really well understood. what we do know is that these systems tend to have lower biomass, a lot more patchy vegetation, and we hypothesized that that would, result in lower surface fuels and really lower the potential for fire spread in these communities. So we would hypothesize that fire frequency in serpentine or forests on serpentine soils would be much less frequent than maybe in the adjacent, more well-developed productive forest types. So to answer this question, we used a technique that I think is so cool and provides this really interesting insight into the past, and that's fire scar dating and fire scar collection. So you can see, for those of you that are unfamiliar with a fire scar, there's a photo here. They're often found at the base of trees, and they're kind of demarcated by this dark black scar. And they occur by low-severity fires, which have occurred in the past, and they actually kill part of the tree's cambium, but don't actually kill the tree, and they leave this wound. And this scar becomes a lot more susceptible to future fire because the bark's been removed. There's increase in pitch, which makes it a lot easier to ignite. So over time, these scars increase as they're burned by subsequent fires. And you can actually date these different fires by looking at the wounds, which are represented as these kind of vertical folds in the blackened wood. And you can take cross-sections from these scars, and you can compare them, the growth rings in those fire-scarred trees, with some of our chronologies that we have for different tree species across the West. And you can get really accurate representations of when those fires occurred in the past and even the season that they occurred. This is a study that Scott Stephens did at Blodgett that just kind of gives you a representation of what these fire scars might look like. They were working at Blodgett and they were creating fire scars on their own using these torches, these small handheld torches. They removed the bark from these trees, burned these trees, and then did them actually at different times of the year. So this photo in the B photograph just shows these multiple scars that were done at different times of the year. And then they let these trees grow for about 3 years, and then they cut them down and looked at their cross-sections. And you can see in the photographs at the bottom, in that C and D photograph, where those fire scars are occurring within those tree rings, and actually how you can kind of get at the seasonality of those fire scars. So in the C photograph, you can see that scar is right at the beginning of that growth ring. So that occurred really early in the season, whereas that D scar was kind of in the middle of that growth ring, indicating that fire actually occurred kind of in the middle of that growing season. So this allows us to not only get really accurate dates of past fires, but also kind of when they were occurring in the past. So we used this technique, these fire scar sampling techniques, to sample trees in our study location, which was in Meadow Valley on the Plumas National Forest. It's situated at about 3,600 feet. And we found a pretty wide number of fire scars within a pretty small area, about a square mile. And we were really interested, again, in finding that ecotone between that kind of non-productive serpentine and that more upland or more productive forest soils. We had 6 sites, and each of them included a pair of serpentine and non-serpentine plots. So we had 3 pairs total. So we used a chainsaw to cut wedges from 57 different fire-scarred trees, stumps, and down logs. We focused on Jeffrey pine, incense cedar, and ponderosa because these are notoriously rot-resistant, so they can record really, really old fires. We puzzle-pieced them together in the field. You can see here us working on one of the cross-sections. We brought the samples back to the lab. The Forest Service has a fire lab in Redding, which does a lot of this fire scar sampling and dating, where they polished and sanded the surface, and then they were able to actually date a lot of those fire scars for us. We cross-dated about 46 samples, which allowed us to produce this really incredible composite fire history for each of our 6 sites. And it's really important, if you're ever going to do fire scar sampling, that you do collections from a number of different trees. Because if you think about it, each tree in that stand will not record every single fire in the past. So it's really nice to get a pretty wide diversity of samples so that you get a pretty accurate picture of how and when fires were burning in the past. So what we found. We had a pretty high number of fires that we were able to record over a pretty long span of time. We found fires operating in these systems over a period of about almost 250 years. The first fire we recorded was in 1650, and the last fire was in 1893. We actually recorded no fires in either of the sites, or in any of our sites, after 1893. And the median fire return interval for all of our sites was pretty frequent. Fires were burning on average every 6 to 22 years in the past, and most of these fires occurred towards the end of the summer or early fall. So I want to walk you through a few of these diagrams, and they're a little complicated, and I'm not good with pointer, but I'm going to try. So these are fire history diagrams, and each of these kind of vertical or horizontal lines represents one of our fire scar samples. And each of these little vertical ticks represents an individual fire. And at the bottom here, or at the top here, you see all of our non-serpentine sites, and here are the paired serpentine plots for each site. And then at the bottom, you have this really nice composite fire history of all of the fires that occurred in both this non-serpentine and serpentine paired site. And at the top, you have these vertical lines, which give you the percentage of all of those samples that were scarred by an individual fire. So I know it's a lot, but we'll walk through it. So what we can tell from this fire history sample at this site is that fire was pretty frequent. And there was very little difference between our serpentine and our non-serpentine sites. The median fire return interval in serpentine was 8 years compared to a median fire return interval in our mixed conifer non-serpentine site of 6 years. So really similar and really frequent. Our second site showed a similar pattern. We had a median fire return interval of 9 years on serpentine and 6 years in non-serpentine. And one of the things I wanted to point out in this diagram were those fires that crossed that boundary. And we had quite a few. And you can see these here, just a few of them that I circled in these orange circles. And that really provides evidence that for a number of years we were having these large fires that crossed the boundary between serpentine and non-serpentine. So you're getting evidence of kind of there being very little barriers to fire spread in these systems. Our third site had a little bit of a longer fire return interval. On the serpentine, we had about 22 years compared to the mixed conifer site, which was about 15 years. And again, we saw evidence of a number of those large fires in those years that kind of crossed that boundary between the serpentine and the non-serpentine. So getting back to our question, what role does fire play in these adjacent but pretty distinct forest ecosystems? I think it's a really interesting question because unlike what we thought, what we found was that fire frequency was somewhat less on serpentine sites than non-serpentine, but fire was still a lot more frequent than we expected. It was burning on average every 8 to 22 years, and there were a number of years where fires would burn from adjacent sites, into serpentine and non-serpentine in a single year. And all of our study sites, regardless of whether they were on serpentine or non-serpentine, experienced this pretty long fire-free period of more than 120 years. And I think that last piece actually provides the insight into why we might actually see these differences. And I thought of this on the drive here, so I didn't prepare a slide, but I was thinking about this the other day, and I really think that if we think about historically what these forests look like, they looked a lot different than what we see today, right? And if you go to some of the talks tomorrow, I know Ryan Tompkins is giving one on some historical stand structure information that you should go to. But what we do know is that at the time when these fires were burning, every 8 to 22 years, the forests that they were burning in were really significantly different. They were much more open, they were much patchier, they were more heterogeneous, they might have had less surface fuels. So at that time, back in the 1600s or 1700s, that distinction between serpentine and non-serpentine may have been very little. It might have all looked like serpentine forest at that time. And it's really when we think about that long period of fire suppression, that long period of 120 years, where we start to see these distinct differences in how these landscapes have responded. So on the serpentine, where you have really slow growth and low productivity, you have a much slower response to that fire-free period than in some of those more productive sites where you're seeing much more densification, much more continuity of vegetation, and that kind of distinct demarcation between those two soil types. So that's my hypothesis I'm working on there for why we see that difference in some of these no difference in some of these fire frequencies. Okay, so management implications. Why does this matter? I think for a number of reasons. I think one thing that could be really interesting is thinking about these things as kind of reference conditions for how forests may have looked in the past. But I also think that we often think of, because they are slow growing and because they are less vulnerable to these long periods of fire exclusion, that we often disregard the effects of fire exclusion in serpentine forests. But I do think that they are changing, and I think that we need to be thinking about reintroducing fire into these systems just like we are thinking about reintroducing them into some more fire-excluded productive forests. So proactive management is likely needed in a lot of these ecosystems, especially if we're interested in maintaining some of the rare and unique plant species that depend on them. And I think that one of the things that is kind of cool from a really practical standpoint as a manager, we can use kind of these estimates from fire history studies like the one we did that really give us these kind of refined estimates of how frequent fires were occurring within these systems, and really allow us to kind of try to develop more refined prescriptions about when and how often we should be burning in these systems. So there are a lot of people that were out cutting fire scars with us and sampling and preparing them in the lab, and I just want to thank all of them, and thank you also for listening.
Am I on now?
Hi, guys.
Thank you for that talk. The observation of what things look like now on and off serpentine versus what things might have looked like 300-400 years back is a powerful one. Mm-hmm. And I think I can have hypotheses along the same line when it comes to grassland systems, where we have now restored serpentine let's stop calling them grasslands. Meadow systems, California prairie lands, where we have serpentine restored ones, and adjacent to them, normal soils that I think look very different, very, very different from what they did in the past. And using that kind of approach of the serpentine giving us a little bit of an insight of the uninvaded, unaltered things that we could be using as a goal to start understanding how fire behaved in the past, because it didn't behave like it is now in those overgrown systems. So it's interesting to see a parallel thought pattern in the meadow systems I work in and the forest systems you work in.
Yeah, absolutely. I think it's a really interesting one. I mean, if you think about it, these systems may have been slower to change, so we can definitely make some parallels. They're imperfect. I mean, they're definitely influenced different species composition, different tree sizes, but I think they do give us some good glimpse into maybe what it might have been in the past.
Thanks. I don't know how good the talk could have been, but it was great as it was.
You don't know Scott Stephens.
Yeah, I know, exactly. So just in the context of what I hear is like very generalized, not knowing specific, but oral tradition that fire was much more frequent than 8 to 22 years in these systems, and that just seeing fire put on the ground by non-CAL FIRE, non-Forest Service practitioners, like, are we missing a lot of the fire return interval by looking at cookies, which is a great tool, but I've just seen so many things where it's just bark scorch, and you're not going to like see that in the system if there aren't a lot of those more like medium size fuels on the ground. Do you think we're just really missing and going on knowledge that just really it can't be complete because of how we're gathering it?
I would be surprised if fire was more frequent than what we're documenting. Every 8 to 9 years is pretty frequent in these systems. So I don't know if I think the fire scar record is definitely imperfect, but I think it is a pretty good proximate for how frequently these systems were burning. Is that what you were asking? Yeah.
Perspective, but that's how it's about.
Yeah, I mean it's definitely imperfect, but it's better than what we have in a lot of other systems, grasslands and things where we have no clues to, except for maybe pollen cores or something, charcoal records. But Yeah.
I was grooving on your batches of cookies there and looking out to the left, which I shouldn't have been doing, but I was, and I realized that the data is less complete. But am I seeing like fewer fires the older the cookies or the
Which one?
were? Yeah, way out on the left, 1550, 1600.
I think that's just a limitation of the trees that we have. Like we were really dependent on having those fire scars preserved and still present. So as you go back to the 1650s, you're getting a little sketchy in terms of what material is actually still present on the stand on site now. So I don't think that we had less frequent fires prior to 1650. I think that's just what we're able to capture in our fire scar record. Does that make sense? Yeah.
I wanted to ask if you had any plans to communicate with the local tribes and ask them about their history of cultural burns and work with them in the management of future cultural fires or prescribed burns?
Absolutely. We do have, one of the things I should have mentioned is bear grass occurs on serpentine in this area, which is a really important basket weaving material for the Maidu. And we've been working with them on experimenting with some prescribed burning to actually promote some of the bear grass materials. But yes, it's a continuing conversation. I mean, just getting the conversation about prescribed fire into some of these systems is just the first step. So yes.
So we're out of time, but since this is the last session, if people do you want people to just come up and talk to you individually?
Sure, whatever. Doesn't matter.
Yeah. Okay, come talk to Michelle if you still have questions. Hey, thanks everyone.